remind to record the meetings. Thank you, Kitusha. Yes, thank you, Kitusha. I forgot to record this part. Yeah, I will provide this recording later on, and some of them are absent today due to a good reason they need again. I get answers, crystal shape and sweet taste, soluble in water, soluble in polar solvent, insoluble in water, right. I think good that I can correct certain things. Okay. Now let's consider glucose. Where do we can see glucose? If we visit hospitals and the person is not taking food for a long time due to vomiting, and when he take the food from mouth, the patient is vomiting. So then it's very difficult to give food through the mouth. And then this person need energy and also fluid. One of the first thing person is admitted to the hospitals, they are connected with the catheter, the cannula, and through the cannula they can insert the tubes to give the liquids. We call it saline. The saline is given. The saline is 0.9% sodium chloride solution. So it is replacing the water, giving the fluid. So we can't give directly water because it's not balanced with the body fluid. So we make the balance with sodium chloride. And people is not eating, they get a glucose. We call it dextrose. So if you look at the dextrose bottle, it is soluble. It is soluble in water. So if you say insoluble, your answer no. Sucrose, the monosaccharide, disaccharide, they are soluble. Monosaccharide and disaccharide, they are soluble. Exception is when it comes to polysaccharide, like starch, they are insoluble. But remember, monosaccharide, disaccharide, they are soluble. Now, if you look at this question, some of them say it's some of them say it's crystal shapes like this. Okay, this question, now look at here. All monosaccharide. I said all monosaccharides are glucose, fructose and galactose. The three of them are reducing sugars. Glucose, galactose, fructose. And here some disaccharides. They didn't say all disaccharide. Why is that? Because reducing power only preserved in maltose and lactose. But sucrose is not reducing sugar. State the common characteristic of all monosaccharide and some saccharides is reducing sugars. They are reducing power. They can reduce. Even this answer is further strengthened by the second part. Describe a simple lab test used to identify sugars with the characteristic of stated as the answer for question 2. Now, if you look at the notes, all the practicals I'm going to do at the end, because otherwise I might miss practicals in the middle of the lesson. So all the practicals I put into the end. So I am doing the practicals at the end. But here, you can remember the test we use to identify reducing sugar is Benedict test. So we can use to identify all sugars using Benedict test, even non-reducing sugars for sucrose. We can use a modified Benedict test to identify sucrose. Modified test. So I will explain you Benedict test for all reducing sugars, Benedict test for sucrose. But here you can remember Benedict test can be used to test the reducing sugars. I think yes, does it? Yes, you added the test and all these things. That's correct, right? Okay, so monosaccharide and disaccharide are soluble, and almost all sugars are reducing except sucrose. We can check in the lab these sugars by Benedict test. Detail of the Benedict test we are going to do at the end of this biological molecules, the practical part, right? Okay, we are moving to the next one, polysaccharides. Right, a poly is anyway, it's a many. Then the many units, many monomers make the polysaccharides. Example, good one is the starch. Now, starch, glucose molecules, they join together by this polymerization reaction. I mean, I can't add all things, I just add like this, like many units like this, glucose molecules join together to make the starch molecules. Now, starch molecules again has two components. One is called amylose. Other one is called amylopectin. If you take a starch, 20% is amylose, 80% is amylopectin. Now what is different? Different is their structure. So amylose molecule, usually it's like this. A long linear, long linear molecules. Amylopectin, they make a branches. They make branches. So how they make the branches? It's like this. Now the glucose molecule join here carbon number 1, and the other glucose molecule react with the carbon number 1 with carbon number 4. So they make the bond. So likewise, many glucose molecules join together to make the linear structure. And another one here carbon number 1. So they can also make a bond with the carbon number 2 of another molecule. This is carbon number 6 and this is carbon number 1. So they have a carbon number 1 6 bond and carbon number 1 4 bonds. So these are the branches. And another thing is the OH groups. The OH group of the glucose molecules, they make a hydrogen bonds with other molecules. So then the OH groups are not available in starch. That means they are engaged with making hydrogen bonds. As a result, it is insoluble. Starch is insoluble. Starch is insoluble. Because if you look glucose, fructose, galactose, they have a large number of OH groups. Even if you take disaccharide, they have a large number of OH groups. These OH groups they have some kind of a polarity. So they can make bonds with the water molecules. Then they are soluble. But starch is insoluble because their OH groups are not freely available. And starch is storage carbohydrate of plants. Storage carbohydrate of plants. You can also copy this down on the polysaccharides. Starch consists of amylose and amylopectin. They are structurally different. Amylopectin is a branched. It's a branched molecule. Amylo is a linear. So OH groups make hydrogen bonds. As a result, they are insoluble. OH groups are not available to react with water. So any question about the lessons so far? We need anything to ask? So far no questions. Okay, I will explain this one again, asked to explain the red color one. What is it? I don't have a space to explain. I have deleted this one. But I will check. Okay. So first you copy this down. Right now, if we take a glucose molecule, this glucose molecule, the oxygen and hydrogen, the hydroxyl group, and they can make a bond with the water molecules, right? So if the water molecule is like this, so this oxygen can make a hydrogen bond. I told you from the beginning, if molecules can make a bond, they are soluble. So most of the sugars are soluble. Like what we discussed, 6 sugars, they all are soluble. Because the OH groups are available. But when we talk about the starch like a big molecule, they make a hydrogen bond between this OH group. So let's say this is the starch molecule. Example of starch molecule. It's a quite a big molecule. So it can make a hydrogen bond. So these OH groups of one glucose make a hydrogen bond with another OH group like this. They're making the hydrogen bonds. So when the hydrogen bonds are made, the OH group is not available in starch. OH groups form hydrogen bonds. So they cannot make new hydrogen bond with water molecules. That's why it is insoluble. Starch is insoluble. It can't make hydrogen bond with water molecule because it already formed hydrogen bond between each glucose molecules. I mean, it is not that important to remember, but you need to remember solubility and insoluble. Why sugars are soluble, why starch is insoluble? Answer is, they can make a bond with water. Glucose can make a bond with water. Disaccharide can make a bond with water. But polysaccharides, they can't make bond with water because hydrogen bonds cannot form as it already formed hydrogen bond between the molecules of starch. Alright, so any other questions? The second polysaccharide is glycogen. First polysaccharide is starch, storage sugar. Second one is a glycogen. Now glycogen, why it is important? It is storage polysaccharide of animals. And also we can add fungi. Animals and fungi, it is a storage source of carbohydrate. So it is made out of, again, glucose. It is similar to amylopectin, but larger. If you remember, amylopectin molecule, they have a lot of branches. Molecule is like this. So it has a 1,4 bonds and 1,6 branches. So different is, it is a larger molecule. So glycogen in human, it is stored in liver and muscle. In humans, stored in liver and muscles. Okay, next is cellulose. Glycogen, starch and cellulose, the three important polysaccharides. So each group, monosaccharide, disaccharide and polysaccharide, you have to remember the important three carbohydrate in each groups. So cellulose, next is the cellulose. So when we take cellulose, I hope you copied this one. Cellulose is the main component of the plant cell wall. This is again composed of the glucose. Now I have a question for you. So one of you can answer. We can eat starch. Human can starch. We can eat starch. So we take main food source is a starch. But we can't use cellulose as our main food source. We can't eat leaves like cows. We can't eat as a main food source to get energy. Question is, if starch made out of glucose, cellulose is also made out of glucose, why we can't take cellulose as a main food source? Right. When you say your answer, most of them, cellulose cannot be digested. Why cellulose cannot digest them? If it is made out of glucose, starch made out of glucose, cellulose made out of glucose, why it cannot digest? Because some of the lack enzymes. Yes, I think when you say cannot digest, it lack enzyme. If we look cellulose structure is different to amylose or amylopectin. It is a glucose, but the glucose carbon number 1, you can see a slight difference. The OH is here. The OH is upward. We call it beta glucose because of this difference. And then next, glucose molecules, it rotates to again 180 degree. This molecule is rotate to 180 degree. And then it make this glycosidic bond. This is completely a different glycosidic bond. We don't have enzyme. We don't enzymes to break. And then it is repeating again like this. Many beta glucose molecule repeating, and the structurally glycosidic bond is different to starch. Enzyme is not, we don't have enzymes to break cellulose glycosidic bond. Only some microorganism, they can make the bonds. This is why some animals like cows, they can eat cellulose, their microorganism can break it. Okay, I think I'm going to stop today with this class. I will update you regarding the next month real classes. I will WhatsApp everything. If you have a question, you can WhatsApp. I will send the link for the recording. Then if you miss something, to copy this note down, or in case some student they don't participate, they can benefit from the records. I will keep update for the next class. Then we can continue from next month. Full power to finish these units, right? If you have any question, please WhatsApp and keep the notes. Then we can meet again next week. We'll again try to find the time as well. I will again post the right date. Okay, thanks, and see you again next